how a tiny cell inside our body decides our eye color, height, and even our face shape. Inside every cell, there is a control system, a factory, a transport network, and even a memory storage. Today, we will understand how cell organelles, DNA, RNA, and chromosomes work together to create life, and why UPSC asks questions from this every year. In the first class, we have understood the structure of cell with the help of city analogy. Let us give it a quick revision and then move forward. So, this is a cell. The outer membrane of cell is known as plasma membrane, whose function is to protect the cell. It is like the boundary of a city. Then, as there is a control system in a city which will control the activities of city, there is the brain of cell, which is known as nucleus. This nucleus decides how the cell will perform its function. Then attached to the nucleus is endoplasmic reticulum. This endoplasmic reticulum is of two types. One is rough endoplasmic reticulum and other is smooth endoplasmic reticulum. The function of rough endoplasmic reticulum is to produce protein because it has ribosomes on its surface, and ribosome is like the protein factory. The function of smooth endoplasmic reticulum is to produce lipid. So, lipid and protein are produced by ribosomes and endoplasmic reticulum. Then the endoplasmic reticulum acts as a transportation system and transport the produced material to the Golgi apparatus. This is known as Golgi apparatus. The function of Golgi apparatus is of packaging. Then, after packaging, these materials are sent to vacuole. Vacuole stores water and nutrients. In the previous class, we have studied the difference between plant cell and animal cell. One of the major difference is the presence of large central vacuole in plant cell, while in animal cell there are many small vacuoles, or sometimes these vacuoles are even absent in the animal cell. Now there is another organelles which act as waste disposal system. It removes waste from the cell, and the energy for all these functions is provided by mitochondria. Mitochondria is known as the powerhouse of cell. So this was the structure of cell. Now let us study cell membrane. Singer and Nicholson gave the fluid mosaic model. Which explain the structure of plasma membrane, and this is the structure of plasma membrane. Plasma membrane is made up of phospholipid bilayer, which means there are two layers of phospholipid. This is one layer of phospholipid and this is another layer of phospholipid. In the phospholipid layer, there are two things. The head is made up of phosphate, while the tail is made up of lipid. The phosphate head is hydrophilic and lipid is hydrophobic. The word hydrophilic is made up of two words. One is hydro, which means water, and philic means loving. And the word hydrophobic is made up of two words. The word hydro means water and phobic means hating, which means the outer phosphate head is water loving and the lipid tail is water hating. इसका मतलब ये है कि जो सेल मेम्ब्रेन है वाटर उसमें से इजीली पास नहीं हो सकता। वैसे तो वाटर के मॉलिक्यूल्स बहुत छोटे होते हैं तो वो पास कर जाता है यहाँ से, बट ये प्रोसेस बहुत ही ज़्यादा स्लो प्रोसेस है। क्यों? क्योंकि बिकॉज जो टेल है वो लिपिड की बनी है और वो क्या है, वाटर हीटिंग है। Apart from lipid and phosphate, the cell membrane also has proteins. There are two types of protein. One is integral protein, which is present across the plasma membrane, and another is peripheral protein, which is present on the periphery of plasma membrane or on the surface of plasma membrane. When the protein are attached with carbohydrate, then they are known as glycoprotein. And when lipid are attached with carbohydrate, they are known as glycolipid. The plasma membrane is quasi-fluid in nature, which means these proteins can move laterally across the cell membrane. It means the cell membrane is not rigid. It is flexible, and this feature is important for cell growth and division. The cell membrane is selectively permeable. Which means cell membrane does not allow all the substances to pass through it. It will select some substances and then only allow to pass through it. There are three methods of transportation. One is active, another is passive, and osmosis. Generally, the substances move from high concentration to low concentration. When the substance has high concentration on one side and low concentration on another side, then the substances flow from high concentration to low concentration. And this is known as concentration gradient. And when the substances move along the concentration gradient, which means when the substances transports from high concentration to low concentration, then there is no requirement of energy, and it is known as passive transportation. And when the substances moves from low concentration to high concentration, then we have to provide energy in the form of ATP to let that molecule transfer from low concentration to high concentration of molecule, and it is known as active transportation. What is osmosis? The movement of water from high concentration to low concentration is known as osmosis. Generally, because there are hydrophobic lipid tail in the plasma membrane, water cannot easily pass through it directly. Because the water molecules are small, they can pass, but this process is very slow. So, for transportation, water passes through aquaporine channels. What are aquaporine channels? These are made up of protein. These are protein channel which will transport water from outside the cell to inside the cell. It will help to transfer water through the hydrophobic lipid layer. This is all about plasma membrane or cell membrane. Now, endosymbiotic theory. There are two organelles, mitochondria and chloroplast. These organelle possesses single circular DNA मॉलिक्यूल। हमने देखा था कि जो जेनेटिक मटेरियल है वो न्यूक्लियस के अंदर प्रेजेंट होता है इन द फॉर्म ऑफ डीएनए और आरएनए, बट न्यूक्लियस के अलावा दो और ऐसी ऑर्गेनेली है सेल के अंदर, माइटोकॉन्ड्रिया और क्लोरोप्लास्ट, जिसके अंदर स्मॉल सिंगल सर्कुलर डीएनए मॉलिक्यूल प्रेजेंट होता है। हमने देखा था कि जो माइटोकॉन्ड्रिया है वो प्लांट और एनिमल सेल दोनों में प्रेजेंट होता है, बट क्लोरोप्लास्ट है वो सिर्फ प्लांट सेल में प्रेजेंट होता है और उसका फंक्शन है फोटोसिंथेसिस। जो एंडोसिम्बायोटिक थ्योरी है वो बताती है कि कुछ ऑर्गेनेलीज हैं यूकैरियोटिक सेल्स के अंदर जो इवॉल्व हुई है फ्रॉम प्रोकैरियोटिक ऑर्गेनिज्म। So, according to endosymbiotic theory, some organelles in eukaryotic cell have originated from prokaryotic organism, especially mitochondria and chloroplast. It is believed that mitochondria and chloroplast have DNA molecule because they have originated from prokaryotic organisms. Recently, nitroplast has been discovered in an algae called Bradysphaeria bigelovi. Recently, Bradysphaeria bigelovi algae के अंदर नाइट्रोप्लास्ट मिला है और नाइट्रोप्लास्ट का फंक्शन क्या है, वो नाइट्रोजन फिक्सेशन कर सकता है। नाइट्रोजन फिक्सेशन विच मीन्स ये एटमॉस्फेयर से नाइट्रोजन लेके उसे नाइट्रेट या अमोनिया में कन्वर्ट कर सकता है। हमें पता है कि जो प्लांट्स हैं वो डायरेक्टली एटमॉस्फेयर से नाइट्रोजन एब्जॉर्ब नहीं कर सकते, उन्हें नाइट्रेट या अमोनिया की फॉर्म में ही नाइट्रोजन को एब्जॉर्ब कर सकते हैं प्लांट। तो इसलिए नाइट्रोजन को नाइट्रेट या अमोनिया में कन्वर्ट करना पड़ता है और इस प्रोसेस को बोलते हैं हम नाइट्रोजन फिक्सेशन। और ये जो फंक्शन है ये मेनली परफॉर्म किया जाता है बाय प्रोकैरियोटिक ऑर्गेनिजम्स, जैसे कि राइजोबियम, एजोटोबैक्टर, ये ऑर्गेनिज्म प्रोकैरियोटिक ऑर्गेनिज्म जैसे कि राइजोबियम, एजोटोबैक्टर, नॉस्टॉक, एनाबिना एक्सेट्रा, इनका काम है नाइट्रोजन फिक्सेशन का। ये एटमॉस्फेयर से नाइट्रोजन लेती है और नाइट्रेट और अमोनिया में कन्वर्ट करती है ताकि प्लांट है वो उन्हें इजीली एब्जॉर्ब कर सकें। अब एक एल्गी के अंदर देखा गया है, कौन सी एल्गी, ब्राडोस्फेरिया बिगेलोवी, इसके अंदर एक ऑर्गेनेली मिली है जिसका नाम है नाइट्रोप्लास्ट और इस नाइट्रोप्लास्ट का काम है नाइट्रोजन फिक्सेशन। तो ये जो है एंडोसिम्बायोटिक थ्योरी को और ज़्यादा स्ट्रॉन्ग बनाता है बिकॉज ये फंक्शन किसका था, प्रोकैरियोटिक ऑर्गेनिज्म का, और अब ये ऑर्गेनेली बनी है। तो एंडोसिम्बायोटिक थ्योरी क्या कहती है कि कुछ ऑर्गेनेलीज हैं यूकैरियोटिक सेल के अंदर वो प्रोकैरियोटिक ऑर्गेनिज्म से मिलकर बनी है और ये एविडेंस उसे और सपोर्ट करता है। नाउ लेट अस मूव टू न्यूक्लियस। This is the structure of nucleus. This is the outer membrane of nucleus, which is known as nuclear envelope. And this membrane is not continuous. There are pores in it, which is known as nuclear pore. Inside the nucleus, there is a dark region, which is known as nucleolus. And the function of nucleolus is the ribosome production. Ribosome production is the function of nucleolus which is present inside the nucleus. Another thing which is present inside the nucleus is the genetic material. This is the genetic material. Genetic material may be in the form of DNA or RNA. Now, what is the characteristic of genetic material? It should be able to replicate, which means it should be able to form its own copies. Second, it should be chemically and structurally stable, which means it should not react easily with the another compounds, and it should be structurally also stable. There should be scope for mutation. Mutation, what is mutation? DNA के अंदर sudden changes. Sudden changes in DNA is known as mutation. अब mutation होना क्यों ज़रूरी है? ताकि वेरिएशन आ सके. म्यूटेशन होगी तभी वेरिएशन आएगी. Another characteristic for a genetic material is expression as Mendelian characters. It means it should be expressed as Mendelian character. It should follow the Mendel's law. So, earlier RNA was the genetic material, but then DNA become the genetic material in higher organism. Why? Because DNA is more chemically and structurally stable, while RNA is not that chemically and structurally stable. It is catalytic in nature, which means it easily reacts with other compounds. Now, we will cover the structure of DNA, then RNA and chromosome in the next class, in the part 2 of this class.